Hey, I'm Mr. Williamson, and this is the third video in the genetics unit. Pretty much going to cover some mutations and a hodgepodge of other things and kind of wrap up the unit. Before we get started, make sure you're taking really good notes. I can't say it enough times. Cornell style works really well for a lot of students. Outline works well. Make sure you cover all of the subjects in the presentation. Don't stop short. But you got to take notes. You do whatever, chews your gum, whatever works for you. Let's get started. We're going to talk about types of mutations. Right here, we're looking at a sentence that I made. The big fat cat ate the wet rat. Notice that all the words are 3 letters long. Why 3 letters? Because that's what a codon is. It's 3 base pairs at a time. So we're talking about DNA, or it could be RNA, but let's just talk about mutations of DNA right now. So each codon is read 3 at a time. The normal DNA would read the big fat cat ate the wet rat. That's what normal would be. Now, there are a type of mutation, like for instance, missense, substitution. We've taken instead of the big, we made it a Z, the biz fat cat ate the wet rat. Well, that doesn't make any sense, all right? And the codons are the same, and each of these codons, whatever amino acids those code for, those will stay the same. But the BIG, the big, becomes biz, and it's only think about a 1 base pair change from a G to a Z, but it does change the entire protein because it changes that one particular amino acid. An example of that would be achondroplasia, dwarfism. We talked about that in one of the earlier videos. Please understand, you will not be responsible for knowing what kind of substitution, what kind of mutation goes with what disease or what disease it is. I'm just giving you examples. I'm trying to show you that these mutations have real effects. All right. The second type of mutation is a nonsense type mutation, where basically we just eliminate one whole bunch of it, or it could be an entire substitution of everything after the big rat. In this case, it's actually what we've done is eliminated fat cat ate the wet, and we just took that part out. It could be that it's been substituted with a whole bunch of nonsense things, but basically, it makes no sense. The whole sentence, the protein or the sentence makes no sense. This would be an example of muscular dystrophy. Muscular dystrophy is something that affects kids. Hawking, Stephen Hawking, the astrophysicist, had it. He was the one in the wheelchair, and he talked into the machine or typed it in. And as you can see, it's got some real crippling things, and basically your muscles wither away. You want to know more about it? Take a look at it. Google it. Google is your friend. All right, another type of mutation is deletion. This is a frame shift type of mutation. All right, what I've done in this case, we've eliminated under the big, I've eliminated the E. So it's no longer the big fat cat ate the wet rat. But you'll notice the only thing missing is the E right here. It still says big fat cat ate the wet rat. But because we've eliminated, we've shifted the frame reading by codons. Remember, we read them by codons 3 at a time. So instead of it becomes this. And it still says the next 3 letters are BIG. After TH, we take out the E, it's still BIG, but we don't read them as those words. We read them in the words that you see here. And none of that makes any sense if you're reading from that table of amino acids and codons. This would, and these were on it, this would not make any sense. And you could read it. You can look at the 3 letters as, this word and this word. These do make no sense at all. So it's a deletion, but that shifts the frame. It shifts the codons one way or the other. An example of this would be cystic fibrosis. Cystic fibrosis is a disease or a disorder where your lungs and your pancreas fill up with this really thick, sticky mucus. And it can be controlled medically with drugs and things, but it does tend to shorten the lifespan of people that have it. But yes, it's as you can see, the airflow is reduced, the lung airways are reduced, and the pancreas, and that's big because the pancreas produces insulin, and insulin helps you take in glucose into your cells and keep you alive. But it's a really thick mucus type thing. The next mutation is an insertion. Instead of a deletion, now we're inserting something. So we have the big, and then I put in a Z. And it's still, read it, it still says fat cat ate the wet rat, but it doesn't make any sense anymore. It's a frame shift mutation because we've shifted the codons 3 at a time. We've shifted them so they don't read the same. All right, it's called a frame. When you shift, when all the bases are in the same place, except for maybe one, maybe we deleted one or we inserted one, that shifts the codon that we're reading, the bases that we're reading at a time. That's called a frame shift mutation. Example is Crohn's disease. Crohn's disease affects your large intestine. I think it's your large intestine. And basically it can be severe, really bad. It gives you some real bad gastric problems. You can see that there's a wrapping of the fat around the intestine. There's fissures that can happen, which causes bleeding. The actual intestinal contents can leak out into your abdomen, which is not good. The wall thickens up, it can actually cause that part of the intestine to die, and in which case they actually have to go in and remove that part of your intestine. I have a couple friends who have had it, or their kids have it, and it can be a very life-threatening, life-altering disorder. Fortunately, they have it under control, so that's good. Duplication, where basically we have replicated the big fat cat ate the wet rat. We've replicated that particular codon. All right, and doesn't happen very often, but it can. But it's an example of this. And I know it looks like it should be Charcot-Marie-Tooth disease. It's actually Charcot-Marie-Tooth or something like that. And basically, I had to look this up. I didn't know what it was. Basically, it's a change in the muscles where it causes, like, one of the big symptoms, and this is the best picture I could find, is a really high arched foot, and it also affects your hands. It can do the same thing to your hands, and it can cause them. It's a crippling disease that changes the structure of the bones and the muscles and the tendons. And then finally, you have an expanding mutation where it just keeps repeating itself over and over again. And this is what occurred during replication of the DNA. And the examples is Huntington's disease. Huntington's disease is very rare. There's less than 200,000 people that get it, that are diagnosed with it in a year. And it causes your brain to atrophy. And it really depends on how many times that repeating of the DNA occurs, actually, I read. The more it occurs, the more pronounced the issue is. But basically, your brain atrophies and you lose cognitive skills and the ability to think clearly and things like that. And because it's genetic, can be passed down. Those are examples of different types of mutations. Understand, I'm not going to ask you about any of this over here. All right, that's not going to be important. I'm not going to ask you for what these are. I might ask you for, if you delete this or if you insert this, is that, what's a frame, which of these is a frame shift mutation? If I remove one or put one in, what happens, the duplication. But basically, understand, mutations are just changes in the DNA that result, remember what DNA does, it codes for protein. It results in mutation results in an altered protein. Then the protein can't do what it's supposed to do if we change the shape of it. So what causes mutations? Well, one, it can happen spontaneously. Right now with COVID, I just read that there are something like 40 confirmed mutations of the COVID virus. And actually, we're going to talk about one of those in the near future in class real quickly. Because the information that you're getting can actually identify, a biology student in my class can identify exactly what probably is happening in that virus DNA or RNA. But it can happen spontaneously. It just changes. Mistakes are made. As a matter of fact, that's how we can tell. They can predict in DNA how often a change occurs in a certain gene, and we can see like this organism has been around for this many years because it has this many changes, and it takes 1,000 years for a normal change to occur. And so if there's 5 changes, maybe that's 5,000 years that organism's been around. Or it could be mutagens. Mutagens are things that cause mutations. Mutagens are not always carcinogens. Carcinogens cause cancer. Mutagens mutate the DNA. Mutagens include things like smoke, nicotine, things like that. There are chemicals like gasoline, acetone, things like that they can cause it. So it can be an environmental, infrared, ultraviolet light is a mutagen. It will alter your DNA. That's why you get skin cancer. But there's radiation in general. X-rays are mutagens. So there's chemical mutagens and there are radiation mutagens. But anything that can alter the DNA. That's why California's the only state that I know that has that. But they have all the warnings. Like the state of California acknowledges that this may cause cancer or something like that on gas pumps and on other things. And even on some packaging, because when you open up the package, the plastic, the vapors, fumes from the plastic that are being released can cause cancer, and they have to tell you that. A little over, but that's what it is. Let's talk about applied genetics. We're into the hodgepodge. Let's finish up a couple subjects. Selective breeding. We have been breeding animals. I talked about this in the very first video. We've been breeding animals since the beginning of history. We know about Asian raising horses. We know about Egypt growing wheat. But we breed dogs. Like I said, there's no dog in the world, no natural dog looks like a Chihuahua. That's an hors d'oeuvre, guys. But we breed them that way because we want some small yappy dog with bug eyes, which I have one. Not mine, it's my roommate's. But it's a dog, yap yap yap, buggy buggy buggy, and up top it off, it's going deaf. So that just makes life fun. We breed farm animals. If you're raising steers for cattle, for beef, you want a cow that that has a lot of beef on it because you're paid by the pound of beef. So if you can get one steer that has more beef than another, that makes you money. Or if you have chickens that lay more eggs, that makes them more profitable. You're growing crops. If you can grow corn that has more ears of corn per plant, per stalk, then that makes it more profitable. You can do that with genetics, and that's what they do. They're constantly looking at ways of adapting the genetics of agriculture to produce either more crops or product or protect the crops or product. Okay, like maybe apply or change genetics to make them resistant to certain pests and things. So that would actually be GMO, genetically modified organism. Some people have a problem with it. Some people don't. Ask me in class, I'll give you my opinion. Recombinant DNA. This is where we can take DNA fragments from another source and put them into an organism. For instance, we can make plants glow because we can take the bioluminescence from, like, say, a jellyfish, that part of the DNA, and we can put that part of the DNA into a plant. And the plant, once it starts growing, will do that. We can... There are other things that we can do, but it's basically taking DNA from one section of a DNA from one organism and putting in another. They actually have grown human ears on the back of mice. How? Take the DNA that codes for the shape of the human ear, put it into the cell of the mouse, and when it's an embryo, we know exactly what cells become what, and we can grow a mouse that has a human ear growing out of it. Now, it can't hear out of the ear, but it has what looks like a human ear growing out of it. And if I could find one, I'll put a picture of it into the presentation when I get there. And then finally, I believe this is finally, cloning. Okay, which is duplicate an organism through the use of DNA. And the way this would work is we would take DNA from one organism, let's say you, because we're going to talk about you in a minute, and cloning. take DNA from you, a cell from you, because all of your cells contain all the DNA. It would really work if it was a stem cell, because stem cells have not become a particular cell, like a liver cell, brain cell, muscle cell, whatever. Brain cell, right, like you've developed those. Not happening. Kidding. Anyways, we take the DNA from that. We put it into a cell that we've removed the DNA from. All right, so we go into another cell. We take the DNA out of that. We put your DNA in, and then we put it into a surrogate mother, and 9 months later, bam, a genetically identical twin to you is born. Now, I've had a lot of students go, that would be awesome, because I would send my clone to school, and I would get to go to the beach. And the problem is, one, it would be 15 years younger than you right now, because it was just born. Number two, it would be the one getting smart, and you would be getting skin cancer at the beach. It's a separate organism. It's not like if it learns something, you learn something. Think about identical twins. That doesn't happen. Okay? It doesn't work that way. Anyways, I think there are some instances where cloning could be really good in agriculture and things. I don't think doing it in humans. I think there could be some real serious issues, both socially and physically, that cloning could cause. But I can see where we might be able to take the DNA from your cell in the future, and let's say you have liver cancer, and so the way we could maybe cure it is by transplanting your liver. But right now, that means you're going to, the rest of your life, you're going to be taking anti-rejection drugs, which suppress your immune system and cause your body to go, okay, well, that's not my liver, but I'm not going to attack it. Well, what if we could put your liver into you, a new your liver? Well, we could grow from your DNA a liver using the DNA codes for all the liver cells, grow a liver, take your cancerous one out, put the new one in, and it would be yours. We didn't grow it on anybody. We grew it in a laboratory, and we put it in. You'd be okay. Your body wouldn't reject it, and the cancer would be gone. That would be the idea. And I honestly think that you will see things like this occurring in your lifetime, because this is a thing, not just cloning, but the use of DNA and manipulation of DNA in cells and things like that are kind of really exciting and taking off right now. So again, let's real quick review. We talked about the different types of mutation, and one of the big things you want to pay attention to is that frame shift, the changing of where the codons are, the frame shift type mutation. We talked about the types of things that cause mutation, whether it's spontaneous or mutagens. We talked about recombinant DNA, where we take DNA from one organism, we kind of some of the DNA from one organism, put it into another one to take care of those things. We talked about applied genetics, where we can then breed things for particular traits that we want, whether it's crops or that stupid little yappy dog with the long hair and the hind legs that are short, the front legs that look like a giant giraffe, and yet it has T-Rex teeth or something. I just made that up. And then we talked about cloning and maybe growing, using the DNA in the future, not to clone whole individuals, but to clone maybe organs and things like that for possible health reasons. I hope you took really good notes. If you have any questions, make sure you ask it in Edpuzzle or in class. Good luck.